Good morning, good afternoon, and to some of you, good evening. I'm Christy Jewell of LabRoots, and I'd like to welcome you to today's live webinar, Unveiling the Role of T-cell Dysfunction in the Development and Progression of Glioblastoma. Today's educational web seminar is brought to you by LabRoots and sponsored by GIBCO by Thermo Fisher Scientific. GIBCO Cell Culture Heroes is a program putting Ph.D. and postdoc researchers around the world in the spotlight. For more information on our sponsor, please visit thermofisher.com/Cell Culture Heroes. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. You can also see at the top of the page multiple tabs. Please click through our related applications, upcoming and on-demand webinars, as well as links to the Cell Culture Heroes webpage. You may be our next Cell Culture Hero. Also, please notice you can share this webinar on your personal social media. Just click on the social sharing tab to let your friends and colleagues know about today's live event. Now, if you have any trouble seeing or hearing this presentation, just click on that support tab found at the top right of your presentation window, or you can let us know you're experiencing a problem by using that Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce our presenter, Dr. Felipe Galvez Cancino, postdoctoral researcher. For a complete biography on Dr. Galvez Cancino, please visit the Biography tab at the top of your screen. Dr. Galvez Cancino, you may now begin your presentation.
Okay, so thank you very much. I'm Felipe Galvez Cancino. I'm a postdoctoral researcher at UCL Cancer Institute. And I'm going to talk about the role of the T-cell dysfunction in glioblastoma. So the topics I'm going to discuss today is basically I'm going to give an overview of the immune response against tumors and the current state of immunotherapy. I'm going to give an overview about our current understanding of the immune microenvironment of glioblastoma. I'm going to share some preliminary data regarding the characterization we have done of the immune microenvironment of one of our mouse models of glioblastoma. And finally, I will discuss our rational approach for the discovery of novel targets for immunotherapy. So when we talk about the immune surveillance against tumors, we have to start by talking about, so we have a tumor, and this tumor has cells that are constantly dying, and these cells can be captured by immature dendritic cells. Yeah, and these immature dendritic cells will capture cells that contains cells and neoantigens that come from cells and mutation-derived proteins. So these dendritic cells will migrate to the draining lymph nodes, and once in the draining lymph nodes, they will present peptides derived from these cells and neoantigens to naive CD4 and CD8 T cells in a three-signal dependent mechanism. So that includes antigen presentation through MHC T cell receptor, co-stimulation, and the production of cytokines. So these T cells will proliferate, and particularly CD8 T cells will become effectors, and they are the main responsibles of killing the tumor. However, the tumor can stop CD8 T cells by using two main molecules. So the tumor can easily express PD-L1, and T cells, as they get activated, they express a molecule that stops their activity called PD-1. And already, like almost 10 years ago, so these studies are already known. So people realized that by blocking this signal with an antibody against PD-1 or with an antibody against PD-L1, so these researchers show amazing results in patients with melanoma tumors with huge remissions of the tumors that associated with infiltration of cytotoxic CD8 T cells. So now another correlation that people found is basically that the amount of mutations of the tumor predict of the efficacy of immunotherapy. And tumors with highest amount of mutations were more responsive to immunotherapy than those with less amount of mutations. And more interestingly, subsequent studies show that the presence of antigens derived from these mutations that could be recognized by T cells predicted the survival of patients with non-small cell lung cancer, melanoma, and other type of tumors, and that these patients that were surviving following these therapies have a huge amount of CD8 T cells in their tumors, and that these CD8 T cells were recognizing mutant peptides derived from proteins in these tumors. So if we want to understand how does a CD8 T cell response work, so we can graph the time versus the amount of CD8 T cells. So we can imagine that in the case of an infection, a vaccination, or even a tumor, so CD8 T cells are in a naive state, and once they recognize their antigen, they start proliferating and they become effectors. And we can talk about memory precursors and effector cells. So once the antigen disappears, so the infection goes away or the tumor goes away, most of these cells die. However, a fraction of these cells survive and become memory T cells. The problem with tumors is that tumors don't go away, and the antigen keeps in the system. And what happens is basically that these CD8 T cells keep recognizing the tumor and they become dysfunctional. So one of the big questions in the field today is how does this dysfunction works? So what we know so far is basically that CD8 T cells recognize the tumor cell, and they recognize these tumor cells in a chronic way. So they are constantly seeing the same antigen, and we have a chronic T cell receptor stimulation. And this chronic T cell receptor stimulation drives several transcription factors that finally result in the increase of PD-1, TIM3, and several inhibitor receptors that their function is basically avoid the overstimulation of these cells that could produce the death of these cells and promote their survival. The problem is that this way to survive drives a dysfunctional state of these cells that avoid them to kill the tumor. And the obvious question of this presentation is what happens in glioblastoma? So when we think of glioblastoma, we are thinking of several types of disease and very complex, with very complex classification. So in very simple words, glioblastoma is a subtype of diffuse gliomas. So basically, any tumor that resembles a glial cell. And there are several types of diffuse gliomas, but the ones we are focusing our research is particularly IDH wild-type glioblastomas, which are the ones with the worst survival of all brain cancers. And when we look inside glioblastomas, we can divide these tumors based on their mutations. And already 10 years ago, several authors classified these tumors in three main subsets based on the type of mutations they have. So we can easily distinguish proneural tumors based in the mutation of P53 and PDGFR, tumors with a classical genotype based on the deletion of CDKN2A and the amplification of EGFR or the expression of EGFRB3, and also a mesenchymal subtype of glioblastoma characterized by the loss of P53 and the loss of NF1. And very interestingly, 10 years ago, it was also in the same paper, it was described that this subset of glioblastoma predicts the response to chemo and radiotherapy in patients. So we can clearly see that classical glioblastomas, so patients that have tumors enriched in this classical genotype tend to respond better to these therapies. So today the obvious question is what could happen with immunotherapy? Will these patients respond the same with this patient, with patients that have enriched one of these subsets will respond better? So, and also for a very long time, it has been believed that glioblastoma has a very poor infiltration of immune cells. So some time ago, so people start looking to the TCGA, to the Cell Cancer Genome Atlas, looking for signatures of immune cells. And actually, there are signatures of immune cells, and what the author found is basically that you can find signatures of CD8 T cells, naive CD4 T cells, regulatory CD4 T cells, macrophages, monocytes, and NK cells, among others. And more recently, authors from taking tumors for single-cell RNA sequencing, they found that there is a small but very frequent amount of T cells in these tumors. So what's the question of glioblastoma is regarding what I've talked here. So basically the question we have and the question we are pursuing is basically, so glioblastoma has T cells, and the question is basically, does T cell dysfunctions occur in glioblastoma, in this type of tumor that have so few mutations compared to others like melanoma and non-small cell lung cancer? And if there is T cell dysfunction in glioblastoma, the next question is, can we modulate this state in order to generate novel immunotherapies for this disease? So we are approaching this problem through a mouse model of this disease. So our mouse model is what we call an orthotopic mouse model, where we can implant glioblastoma cells in the brain of mice, and 3 weeks later we can analyze these tumors by flow cytometry. So the characteristics of this mouse model that we have developed and analyzed in collaboration with Steve Pollard in the University of Edinburgh is basically that this cell line is derived from neural stem cells, and it carries the same mutations as the human disease. We are working with a model that it is NF1 knockout, PTEN knockout, and it overexpresses EGFRB3. And the cool thing about this model is that we can track this model in vivo, and if we get the brain, we can also see the tumor under the microscope, which allow us to cut the tumor and analyze specifically the immune microenvironment inside this tissue. So what we have seen is basically if we analyze the brain of a mouse without a tumor, we can see, as it has been described, that a normal brain is dominated by microglia. In this dimension reduction called UMAP, where we can observe all the immune populations that reside in a normal brain, we can see in purple that the main population that resides in a normal brain are microglia. So, but the question is what happens in our mouse model of glioblastoma. So if we cut specifically the tumor, we can see that the glioblastoma tumor is enriched on myeloid cells, particularly macrophages. And also it has T cell population, characterized by the infiltration of CD4s and CD8 T cells. And if we quantify the proportion of these cells, and we look first at the innate compartment that comprise myeloid cells and NK cells, we can see that in the tumor, it looks like microglia tend to disappear, but we believe, we haven't done a deep characterization of this, we believe that it is more like microglia tend to change their phenotype, and they tend to look more like macrophages. And we can see that macrophages are increased and NK cells are also increased inside the tumor. And when we look at the T cell compartment, we can clearly see that there is an increase on CD4 effector T cells, CD4 Tregs, and CD8 T cells. Yeah? So we see that there is an infiltration of several types of cells in this tumor. So the next question we ask is, what's the phenotype of these cells? So we perform a differential state analysis of these three groups. So we compare the tumor against the control brain and the non-involved tissue, which is the healthy tissues surrounding the tumor. And what we can see very clearly is that the cells that accumulate in a significant way inside the tumor is basically macrophages. And also we can clearly see subset of CD8 and CD4 T cells, which are PD-1 positive, which is a marker of, as I already explained you, of dysfunction, or T cells which are activated. And these cells are PD-1 positive, Ki67 negative. So it means that they are probably tumor reactive, or for some way we don't understand, they are expressing PD-1, and they are not proliferating. And we can clearly see that neither CD8 or CD4s are proliferating in our model. And when we quantify this, we can, by looking at PD-1 versus Ki67, we can clearly see an increase on cells which are PD-1 positive, Ki67 negative, suggesting that for some reason we still are trying to understand, these cells accumulate in the tumor, they express PD-1, but they stop proliferating. So they are probably entering in a quiescent or dysfunctional state. And more interestingly, when we look at the expression of other markers of exhaustion, classic markers of exhaustion such as LAG3, TIM3, we can see that most of these cells are negative for these markers. So what we believe is that in glioblastoma, T cells enter in a very particular state of dysfunction that could be different from what we have seen in other types of tumors. And in order to validate our results, we went into human data and we look at the public data sets. So we look at a data set of several types of tumors that was recently published in Nature Medicine in January of this year. So we took this data, we reanalyzed it, and for example, if we look at renal cell carcinoma and non-small cell lung cancer, we can see that most of the T cells are in a state where they are PD-1 and Ki67 double positive, which means that they are exhausted, or they are recognizing their antigen in the tumor, but they are proliferating. So now what happens if we look at glioblastoma? What we can see is that this population that was very abundant in renal cell carcinoma and lung cancer, which was this PD-1, Ki67 double positive population, it is not that abundant in glioblastoma. And as it was in our mouse model, the population that tend to be more abundant is this population of PD-1 positive, Ki67 negative cells. So now the question we are pursuing in the lab is basically how can we reverse this dysfunctional state of T cells? And that's the question that many people is asking today. So we believe that one way to do this is using gene editing by CRISPR. So as you might know, CRISPR-Cas9 is a gene editing technique that allows us to very easily target a gene and knock out this gene in any type of cell. So very recently, people have done this on T cells. So this is from the group of a group in New York. They took T cells. They did something beautiful. They took these T cells. They redirect these T cells against a tumor antigen, and they knock out PD-1, and then they reinjected these T cells into patients with cancer. So this was the first study using gene-edited immune cells to treat cancer. Okay, this was a very small study without positive results, but it was groundbreaking because it was the first study to do this in human cells and to apply it to patients. Yeah, so basically, but we are interested in discovering new regulators of T cell function and use this to avoid T cell dysfunction, to recover this dysfunction state that T cells undergo in the tumor. So how do people discover new targets? So what people does and what everybody in the field is doing, they usually take human T cells that can be tumor-infiltrated lymphocytes, peripheral blood mononuclear cells. They do CRISPR-Cas9 on those cells. They knock out any gene they think that could be relevant, and then they do a functional assay. So in that functional assay are usually assays where they make the T cells proliferate or recognize a tumor as they think it could be in the human tissue. Then they analyze this by single-cell RNA sequencing, and they identify which gene could be relevant. The problem with this approach that we believe is not good at all is basically that everybody is discovering the same targets. So what we believe is that this classic approach of discovering novel regulators of T cell function leads to the identification of assay-dependent genes. So as people use functional assays that are related to cell proliferation, most of the people discover the same genes related to cell proliferation. So thinking of this, we've taken a more rational approach, and what we have done, so this is data from lung cancer that we are trying to apply to glioblastoma. So what we have done is doing single-cell RNA sequencing from lung cancer tumors. So from lung tumors, we analyze from T cells in these tumors, we analyze the transcriptome of these T cells, and then we specifically look for genes that could be driving this dysfunction state on T cells. And what we expect is that this approach, this more rational approach, could lead to a better discovery of targets for immunotherapy that can be used to reverse T cell dysfunction. So, and I want to show you just one example of our rational approach and how powerful it can be. So one of our targets, which is a regulator of the T cell receptor, a negative regulator of the T cell receptor, what we have seen is basically that when we knock out this target, we can see a huge increase in the production of interferon gamma on CD4s and CD8 T cells. So just by using this approach and just by targeting one of our more than 100 targets that we have identified by using single-cell RNA sequencing, we can already find a molecule that can be used to reverse a dysfunctional state on T cells. So to finish, I would like to give some conclusions. So basically, I'd like to say that glioblastoma, due to its impressive capacity to thwart the immune response, represents a huge opportunity to understand the underlying mechanisms of T cell dysfunction. Also, I would like to say that CRISPR-mediated gene editing will allow us to genetically modify T cells and make them resistant to dysfunction. And this definitely holds promise for the development of novel adoptive cell therapies for glioblastoma. And finally, what I would like to say is that immunotherapy holds promise for treating glioblastoma. It hasn't been successful yet, but we are sure that by targeting T cell dysfunction and or myeloid cells, it could be the next generation of immunotherapies that will definitely work for glioblastoma. So finally, I would like to acknowledge to my laboratory, to my mentor, Sergio Quesada, and to my collaborators in Edinburgh and here in London. Thank you very much.
Thank you, Dr. Galvez Cancino, for your informative presentation. We will now start the live Q&A portion of our webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions already coming in. Let's start with this one. Why do you think that T cells stop proliferating in the tumor microenvironment?
Okay, thank you for the question. So what we believe is this is probably something very unique of glioblastoma tumors, that more than T cells becoming exhausted in the tumor microenvironment, they are likely to stop proliferating. And we believe that they tend to stop proliferating and because due to the lack of resources that they need to proliferate. So one of the things we are exploring right now, and that can be eventually interesting as a therapy, is basically using, so we believe that probably the lack of interleukin-2, which is key for the proliferation of T cells, of the lack of IL-7 and IL-15, is probably driving this loss of proliferation on T cells. So what we believe is basically that if we can in some way give IL-2 in a way that is not toxic, or maybe also give IL-7 and IL-15, which are cytokines relevant for T cell proliferation and the generation of memory T cells, could be a way to generate like new therapies for glioblastoma and recover this dysfunctional state of T cells.
Thank you. Now, Dr. Galvez Cancino, how do you plan to use gene-edited T cells for treating glioblastoma?
Yeah, so that's particularly challenging nowadays, but what people is doing and what we believe it will be the future of these therapies is trying to take T cells from the tumor of the patients, expand these T cells in vitro, and then edit these cells in the laboratory, and then reinfuse these T cells into the patients. This is something that has worked already in tumors like melanoma and lung cancer. It's called adoptive T cell therapy, and that's a way to use gene-edited T cells for treating for eventually treating glioblastoma in tumor. The other way to use gene-edited T cells could be using CAR T cells. That is known that these CAR T cells actually give some benefit to patients with glioblastoma, and we could edit these CAR T cells and reinfuse them into patients. That's something that probably there is people doing it. Yeah, and what we plan is to use one of these strategies, but using our targets that we have discovered.
Which molecule do you think is most promising about T cell dysfunction?
Okay, so that depends. So molecule for like as a therapy, I think there is a lot of molecules that are very promising. But I think what we have to look for is a molecule that can avoid T cell dysfunction without making the cells die. For example, recently there was described a transcription factor that drives T cell dysfunction, but when the authors of the study knock out that transcription factor, all the cells die. So in some way, T cell dysfunction is needed because it avoids the cells to become overactivated and die. So what we need is, for example, looking for a molecule like a negative regulator of the T cell receptor. So if we can make the T cell receptor more sensitive to antigen stimulation, and that drive like T cell activation in a more physiological way, could be a good target. That's something that has been also recently published. For example, Nicolas Restifo last week published a paper analyzing several regulators of the T cell receptor, and they found that if you can knock out one of those, I think it was P38, you can make T cells that have a memory phenotype. They are high effector, and they do not differentiate and become like terminally differentiate quickly.
Thank you. Have you looked at macrophages status in TME?
Yeah, we are doing that. So we are using this orthotopic mouse model I just showed to look at that, and we are also using something we call de novo tumor models, where the tumor develops very slowly in a more physiological way. And we have looked at macrophages. They look very interesting. We are also interested on developing therapies targeting macrophages. And it makes sense, it makes a lot of sense with what have recently described by Volker Becher and Joanna Joyce from Switzerland, where they found that there is a big heterogeneity of macrophages, and both microglia and monocyte-derived macrophages reside in glioblastoma tumors. What I think is relevant and what will drive our research is basically trying to identify how does the macrophage and T cell phenotype changes in each subtype of IDH wild-type glioblastomas, mesenchymal, classical, and preneurons.
Thank you. Now we have time for a few more questions, but I want to let our audience know that any questions we're unable to answer today, and additionally, those questions that come in during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Okay, let's continue. How important is T-cell differentiation in glioblastoma? Is this related to the different subtypes of glioblastoma?
Yeah, that's something we don't know. Yeah, that's something it will be very interesting to do research about that. So what we know is basically papers that have been recently published. A couple of weeks ago, they basically say that, for example, if we compare glioblastomas which are IDH wild type, which are the ones I talk about it, versus glioblastomas which are IDH mutant, which are a different subset of glioblastoma that tend to respond better to some therapies, they are actually very different, and the T cell status is very, very different. But what are the determinants of this is totally unknown until today.
Thank you. Now, Dr. Galvez Cancino, I'd like to get your opinion on this. What do you think will be the next generation of immunotherapies for glioblastoma? Against which molecular targets will these therapies maybe be directed?
Okay, yeah, that's a question we are asking every day. So what we believe is basically glioblastomas are highly infiltrated by myeloid cells. So macrophages, monocytes, neutrophils, etc. So what we believe, and today most of the immunotherapies that are being successful in the clinic have been targeting T cells. So if we want to develop novel immunotherapies for glioblastoma, we have to move beyond T cells probably, and find a way to make a space to T cells by eliminating other type of cells, like myeloid cells, where if we target macrophages or monocytes with an antibody that can deplete these cells, or by targeting directly the tumor cells and trying to, in some way, deplete these tumor cells.
Thank you. And we have time for one more question. What are the determinant of the immune infiltration in glioblastoma? How does the tumor mutations play a role?
Yeah, that's also very interesting. So we know very few about that. So what we know is from very old studies that, for example, one of the most common mutations in glioblastoma is the loss of NF1, which is a key driver of the mesenchymal subtype of glioblastoma. And for example, just by not having NF1, so glioblastoma cells can recruit macrophages. That's a very clear example of how a driver mutation can be controlling the tumor microenvironment. Regarding other type of mutations, there's very few that we know. So we hypothesize that, for example, in the case of EGFRP3, it could be driving a T cell response just because it acts as a neoantigen, because it comes from deletion in the EGFR locus that originates a new protein that there's no T cell tolerance, so it acts as a neoantigen. But regarding like all the other mutations, we know very few. It's not clear how do they influence the immune infiltration of these tumors.
Thank you, Dr. Galvez Cancino, and thank you for your time today and for your important research. Do you have any final comments for our audience?
Yeah, no, I would like to acknowledge all the audience. I would like to acknowledge GIBCO and Thermo Fisher for the opportunity, also LabRoots for organizing. Any other questions, I'm happy to receive emails, or people can follow me in Twitter. It's Galvez-Cancino. And I'm happy to answer any other question by this media. Thank you very much.
And thank you again. And before we go, I would like to thank the audience also for joining us today and for their interesting questions. As a reminder, those questions we were unable to answer today and those submitted during the on-demand period, they will be addressed by Dr. Galvez Cancino via the email address you provided at the time of registration. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon. Have a great day.